Knotweed Lab here. Today I'm going to walk you through an actual DNA extraction. Stay tuned. A bit of background so you understand this video. We are extracting DNA. DNA is a self-replicating material that is present in nearly all living organisms. Studying it will tell us a lot about where knotweed is coming from and how it's spreading along the river. However, DNA is very small and therefore at the microscopic level, and extracting it can be difficult and hard to understand. A microcentrifuge tube, which is shown here, is a small cylindrical plastic container that allows one to centrifuge small amounts of liquid. Centrifuging means to separate a liquid into two different phases or components. For DNA, it also helps to use spin columns, which are shown here. In this scenario, the mesh will hold the DNA or the precipitate, and the supernatant will be expelled through the mesh into the collection tube. Now that you've been educated on DNA extraction, let's begin with the video. The leaf is first emulsified in the mortar and pestle and then transferred into a microcentrifuge tube with lysis buffer A. The lysis buffer A is formulated to break open plant cell walls and release the cellular contents, including DNA. Another buffer, lysis buffer B, is then added, which has a similar function but is still used to optimize cellular content extraction and inhibit any enzymes that may degrade the DNA. Then, RNase A will cleave single-stranded RNA to fragment it. Removing the RNA is vital to ensure the purity of the extracted DNA. After incubation, a precipitation solution is added to remove proteins, which are then centrifuged out, leaving the supernatant to contain the DNA. Then, we add a binding solution. It contains concentrated salts and alcohol groups that will reduce the solubility of DNA in the aqueous solution. Centrifuging will remove other unwanted cell contents, allowing the DNA to precipitate out. However, because the DNA is so microscopic, we use a spin column which will suspend the DNA but allow aqueous liquid to pass through. And our goal of the DNA is in the mesh you see here. You can see how green the supernatant looks, probably a lot of chloroplasts collected. Then we use what we call the wash, which is a solvent that will ensure that any contaminants such as proteins or enzymes are completely removed. And finally, we add the elution buffer, which releases the DNA from the mesh. Then we can take a drop of our highly concentrated DNA and read it on a machine that tells us our concentration. And that's it, except for dishware. It's a lot of cleaning. I feel like being a researcher is often being a glorified dishwasher.